Group 1 will be presenting a paper entitled Orientation of Asymmetric Stem Cell Division by the APC Tumor Suppression and Centrosome. Hi, my name is Alyssa, and I'm going to be doing the introduction for this paper. And first, I would like to start with a discussion about stem cells and symmetric versus asymmetric stem cell division. Stem cells are cells that have the potential to develop into many different types of cells within the body. Stem cells are different from other cells in three main ways. One, they divide and renew themselves over a long time. Two, they are unspecialized, meaning they don't have a specific function in the body. And three, they have the potential to become specialized cells, such as somatic or gamete cells. There are two main types of stem cells, embryonic stem cells and adult stem cells, but today we're focusing on adult stem cells. Adult stem cells maintain populations of highly differentiated but short-lived cells, such as skin and sperm cells, and maintain a critical balance between alternate fates, whether the daughter cell maintains a stem cell identity via proliferation event or initiates a differentiation into a somatic or gamete cell. Symmetric division, it occurs in two different ways via proliferation or differentiation. Via the proliferation method, a stem cell divides and produces two daughter cells of the same type, so both being stem cells. In the differentiation method, a stem cell divides and produces two daughter cells of the same type, both being non-stem cells, so either being somatic or gamete cells. In asymmetric division, which is the main focus for today, a stem cell divides and produces two different types of daughter cells, one being a stem cell and one being a differentiated cell, which is either a somatic or a gamete cell. Sorry, it's taking a long time to load. Okay, apologize about that. Skipped. Okay, so the model organism for this paper is a Drosophila, and the main content being covered and investigated is male Drosophila asymmetric germline stem cell division, GSC division. In this first image here, it is a up-close view of the apical tip of the male Drosophila testis. So starting from left to right, the hub represented in the blue cells is a cluster of somatic cells at the testis apical tip and functions as a stem cell niche. The stem cell niche is an area of tissue that provides a specific microenvironment in which stem cells present in undifferentiated and a self-renewal state. Cells of the stem cell niche interact with stem cells to maintain themselves via a proliferation event, self-renewal event, or promote their differentiation into, in this case, gamete cells, since we're talking about germline stem cells, specifically sperm cells in this case. In Drosophila testes, germline stem cells, GSCs, represented in green in this diagram, normally divide asymmetrically, giving rise to one stem cell and one gonioblast. The gonioblast is represented in this image by the gradiently colored cell labeled as a GSC daughter cell, and it's an immature stem cell which initiates a differentiation starting with a spermatogeneal transient amplification division, which is represented by the yellow cells in this diagram. The second diagram that appears before you is a schematic of the mechanisms that allow this GSC asymmetric division process to occur. The apical hub cells, which is purple in this diagram, express the signal ligand unpaired UPD, which activates the Janus kinase signal transducers and activators of transcription of the JAK-STAT pathway within the GSC cell, which is represented in yellow in this diagram, to maintain stem cell identity via a proliferation event. The AJ in the blue oval stands for adherent junctions, and in this case, it's adherent. And then the pink structure in this diagram labeled APC2 is one of two Drosophila homologs of the mammalian tumor suppressor gene. This APC2 is located here next to the AJ, known as the ecadherin, and between the hub and GSC interface, since it is an anchoring platform for localized concentration of APC2. The main research question of this article was, what is the relationship between the niche and the mechanisms that ensure the correct balance between male Drosophila germline stem cell self-renewal and differentiation?
Hi everyone, I'm Narad. I will be presenting figure 1, where the researchers show that male GSCs orient toward the hub throughout the cell cycle. So for this part, the researchers analyzed male GSCs, dividing male GMCs in early germ cells using GFP alpha tubulin, antibody to gamma tubulin, antibody to alpha spectrin, and antibody to spectrosomin. And you'll see what each of those was used to label as I go through the panels. So they find that in 100% of the cells that they looked at, the mitotic spindle was oriented perpendicular to the hub GSC interface, with one spindle pole located within the crescent where the GSC contacted the hub. You can see that in A and B. In C, they show that in gonioplasts, which are immature stem cells, the spindle pole, the mitotic spindle, was not oriented toward the hub. And D through F, they look specifically at centrosomes. So the single centrosome seen in early interphase was seen located adjacent to the hub. And after centrosome duplication takes place, the one centrosome remains adjacent to the hub, while the other one migrates to the other side of the nucleus. So you can see that in D, in E, and then in F. And the percentages seen in F are referred to the approximate percentage of cells observed that indicated that centrosome position. And next, the researchers believe that the mechanism for Drosophila GSC spindle orientation was different between sexes. In female GSCs, there's an intracellular spherical and membranous structure called the spectrosome that is localized next to at the apical capsule, where it may aid in anchoring the mitotic spindle during mitosis. But in panel G, in interphase male GSCs, the researchers see that the spectrosome is often located to the side, while one centrosome holds that stereotype position adjacent to the hub.
Hello everyone, my name is Nick Mathias and I'll be presenting figure 2. The researchers were analyzing the centrosome's involvement in asymmetric stem cell division at the germ stem cell hub. To do this, they introduced knockout of centrosomin, the central centrosome component, and they witnessed a disorientation of the mitotic spindle causing malfunction in asymmetric stem cell division. Figure 1A and B show the correct conformation of the mitotic spindle, in which it is perpendicular to the germ stem cell hub interface. Figure 2A and B show misoriented spindle, in which it is parallel to the germ stem cell hub interface, and B, which a germ stem cell separates from the hub. Figure D shows a misalignment of the centrosome, stained in red, as neither of these are located at the germ stem cell hub interface. Figure 2C represents the proportion of centrosomic misorientation with respect to cell phase. The highest rate of misorientation was found to be at metaphase, which makes complete sense if you think about the role of the centrosome. Figure 2E represents the frequency of germ stem cells with mispositioned centrosomes according to the conformation of the centrosomes. The normal diametrically opposed centrosomes aligning perpendicularly with the hub GSC interface decreased by about half. Two conformations were shown that were not observed in wild type before. One, the misoriented centrosomes increased quite a bit, where neither centrosome were adjacent to the germ stem cell hub interface. Figure 2K and 2B illustrate two essential failures of the asymmetric division. K shows the parallel division, and B shows a detachment of the germ stem cell from the hub. Symmetric cell division route 1, as I'll call it, where you have the parallel division, results in both daughter cells remaining within reach of the unpaired stem cell renewal signal, and thus both retain stem cell identity. Route 2 shows the detachment of germ stem cell from the spindle, in which case both daughter cells lose the renewal signal unpaired and differentiate into gonioblasts. Figure 2L further illustrates this, that this is not an artifact, with the time-lapse imaging showing the actual detachment of a germ stem cell from the hub. Figure 2G through J show an increased germ stem cell population and altered morphology at the hub. G and H show the wild type conformation and division, where I and J respectively show the CNN knockouts, overpopulation, and morphological changes. Figure 2F quantitates the germ stem cell population at the hub, where the CNN knockout showed an increase in average opposed to the wild type. There was no observed significant difference in hub size, and therefore the conclusion was made that this increase in population was due to symmetric division, where both daughter cells receive self-renewal signals.
Hello, my name is Jack Fitzgerald. I'm a student in the MSSE program here at UAlbany, and I'll be talking about Figure 3, how APC tumor suppressor homologs are required for spindle orientations in male GSCs. So the way that these APC tumor suppressor homologs are involved is that they are anchored by two adhesive-like proteins. They are called DE-cadherin and armadillo, and these co-localize at the hub GSC interface. In Figure 3E through H, it demonstrates how APC2, which is a Drosophila homolog of APC, a human tumor suppressor gene, they can help anchor astral microtubules to orient centrosomes and the spindle. Now, in APC2 mutant males, there were GSCs with mispositioned centromeres, misoriented spindles, or detached spindles, and it could be a combination of those three. Also, GSCs did not appear crowded around the hub like in CNN mutants, and that's possibly due to an enlarged hub. So as you can see right here, the spindle fibers and centromeres are not oriented correctly toward the hub. Now in Figure 3J and K, that goes over the other Drosophila APC homolog, APC1. So its role in wild types is that it can contribute to normal orientation of the centrosome and spindle during interphase. It localizes to centrosomes and spermatogonia during the late G2 phase during the cell cycle and around the start of prophase. However, it's not detected after the start of prophase. So once prometaphase begins, it's no longer detected. And in APC1 mutant males, the results reported a change in GSCs in spindle orientation and centrosome position, and also the number of stem cells per testis and hub size was also larger when compared to wild types. Now in Figure 3L, it shows a very detailed model for asymmetric cell division in Drosophila male GSCs. As you can see right here, there's the hub. It's a little blurry, but here's the armadillo and the DE-cadherin orienting with APC2 along GSC and properly orienting the centrosomes. So these results propose that asymmetric outcome of GSC divisions is due to extrinsic factors that identify stem cells and an intrinsic factor that orients the cell division plane based on the microenvironment. Also, astral microtubules can be captured by protein complexes at the GSC cortex that interfaces with the hub.
Hi, my name is Maddie, and I'll be presenting the conclusions. So basically, there are three main takeaways from this paper. One, that male GSC is oriented to the hub, toward the hub. Two, that centrosome is important for proper spindle orientation. And three, APC is also necessary for proper spindle orientation. And this diagram over here is just showing basically the two different fates that a germline stem cell can have. It can either divide symmetrically or asymmetrically, which is what we want to see. One stays at the hub and one goes on to differentiate. So focusing on the first result, male GSCs will divide via asymmetric division. Basically, that means that one will stay toward the hub, highlighted there on the black box, and one agonial goes away from the niche in order to differentiate there in the red box. And this diagram down here is just showing an overview of the diagram on the right. The second result shows that the no mutants for centrosomin have misoriented spindles and centrosomes. So in a normal germline stem cell, one will stay at the hub and one will go on to differentiate, like I mentioned. So this is what we would want to see in the germline stem cells. Again, here is a diagram showing proper alignment of the centrosomes and then therefore proper spindle formation and asymmetric division, whereas here the centrosomes are not aligned with the hub and they cannot go on to asymmetrically divide. And just one more diagram showing again, here is the proper oriented spindle showing that it will lead to asymmetric stem cell division versus a misoriented spindle. This leads to symmetric stem cell division, which is not what we would like to see. And the third result is that APC homologs were also required for proper spindle orientation. So APC mutants show that the spindles were either mispositioned, misoriented, or detached, which leads to improper division. So this diagram here is just showing the changes throughout the cell cycle. Here, the centrosomes are at two different poles of the cell, leading to one self-renewal germline stem cell staying at the hub and the other going on to become a gonioblast and differentiate. Over here, again, is another diagram showing a wild type versus the APC mutant. And here, APC is present. You can see the centrosomes are aligned, the spindle is aligned. This cell will go on to asymmetrically divide, whereas over here, with the APC being absent, the spindles and centrosomes are not properly oriented with this cell here, and they will not go on to asymmetrically divide. So the significance of these results is that it shows the importance of the centrosome and the spindle in proper asymmetric division. The authors theorize that APC tumor suppressor is the recruiting factor for this proper asymmetric division, since when it's removed, the spindles were not aligned correctly with the hub. They also see that stem cell orientation is critical for reliable asymmetric division. If the stem cells were not aligned properly, they will not asymmetrically divide. And the authors believe that this mechanism could be a general feature of other stem cell systems. So I think that this could be a future direction for more research in the future, seeing if this mechanism is responsible for maintaining the balance between stem cell self-renewal and differentiation in other systems and other organisms potentially in the future. These are the references that we used, and thank you for watching.